So trisomy X is a genetic variation, an X and Y chromosome variation. And in talking about genetic counseling today, I'm going to also talk about the context of understanding trisomy X in comparison and alongside some of the other variations as well. So to begin with, in talking a little bit about terminology, chromosomes are the structures that carry our DNA. And so our DNA encodes our genes, and our genes tell our bodies how to grow and how to develop. When we use the term aneuploidy, we're talking about the presence of either an additional or a missing chromosome in the cell. And when we talk about sex chromosome aneuploidies, or SCAs, it's specific to having either an additional or missing X or Y chromosome. When we use the term sex chromosome trisomies, we're talking about having 3 copies of a sex chromosome, or the sex chromosomes. So the common conditions that we refer to are Klinefelter syndrome, or XXY, Jacob syndrome, or XYY, and trisomy X syndrome, or XXX. And while the classification of these conditions kind of as an umbrella term, there's a lot of different terms or names that are used, but for the most part, they really are all the same from a classification perspective. So scientifically, in a lot of the medical journals, you'll read about sex chromosome aneuploidies, which is a more kind of medical jargon or scientific term. But sometimes people will refer to these conditions as being sex chromosome abnormalities, anomalies, disorders, variations. And then there also is terminology that's used as X and Y chromosome aneuploidy, or X and Y chromosome variations, that I found that many families often prefer in using that term, as it is fairly self-evident in what it is. When we talk about X and Y variations, there's a lot of different types of X and Y variations that can be classified under kind of this umbrella. So we're going to talk about trisomy X as one of these conditions, but just keeping in mind that there's not only trisomy conditions where we see 3 copies of the sex chromosomes, but we can also have other conditions that are represented on this slide in regard to having additional or missing X and Y chromosomes. And together, combined, when we're talking about sex chromosome aneuploidies, they're really common. They occur in about 1 in 400 live births. We'll see that the most common of the SCAs is Klinefelter syndrome, being 1 in 650 males, but trisomy X occurs in approximately 1 in 1,000 females, as does XYY. So it's a relatively common condition that we can see in the general population. From a history and background perspective, in 1956, Dr. Patricia Jacobs identified chromosomes for the first time under the microscope. And so this was identifying that the presence of the extra X chromosome was in individuals associated with various medical and physical differences. And in the 1970s, we started learning a lot more from newborn screening studies that were initiated and understanding the variability that we can see in trisomy X, as well as the impact that having that extra X chromosome can have. Today, if you search through PubMed in the medical literature, you'll find a total of about 622 articles talking about trisomy X. And relative to other medical conditions, especially in context of how common trisomy X is, this is very small. There's a lot, thousands and thousands of medical journal articles that talk about other genetic conditions. And so trisomy X relative to those is really understudied. The highest year that published about trisomy X was 2021, with 35 publications that were published that year in medical journals. So we really have a lot of work to do in trying to support research in this condition and learn a lot more. When we talk about trisomy X and how it's identified, we know that historically only about 10% of women with this condition were identified during their lifetime. So the majority of what we've learned about trisomy X historically really is based on a small percentage, that the majority of individuals go undiagnosed. And of those individuals, why they were identified, it often is attributed from a postnatal perspective, either to presenting with global developmental delays, hypotonia or low muscle tone, or autism, as these are traditional indications to order genetic testing. But what we know about trisomy X is some of the more common features like ADHD or anxiety, while they're present in a lot of individuals with trisomy X, these diagnoses don't necessarily trigger genetic testing to be ordered from the physician caring for these patients. So today, we are identifying a lot more babies with trisomy X. And this is due to the initiation of non-invasive prenatal screening. So this is a maternal blood test. So it's non-invasive, meaning that there's no risk to the pregnancy and resulting in miscarriage from the procedure. And it's a test that's looking at fetal DNA fragments that are shed from the placenta in the developing pregnancy into the maternal blood. And so it's testing for these fetal DNA fragments in combination with the maternal DNA fragments to be able to see if there's an increased risk for having trisomy X or other chromosome abnormalities. So it's a test that's also commonly referred to as cell-free DNA testing. And it's now in 2020, as of 2020, recommended that all women, regardless of maternal age, be offered this type of screening. And this is from the American College of Obstetrics and Gynecology. It's a test that's typically initiated at 10 weeks or later in pregnancy, and that's because we need the fetal fraction or the concentration of the fetal fragments to be high enough to be able to successfully identify these conditions during pregnancy. The results from the screening test can be positive or an increased risk for trisomy X. They can be negative, in which they don't identify an increased risk, or there can be inconclusive. And there's a lot of variable methodologies that the labs are using. So not all labs are created equal from a technology perspective. In a recent publication from the American College of Medical Genetics, when they did a meta-analysis of 28 studies about the positive predictive value or the likelihood of a positive screening result being a true positive result or diagnostic result in the individual or the fetus, they found that the positive predictive value for trisomy X was a little more than half. So it's really important that if there is a positive non-invasive prenatal screen result, that it be followed up at some point with diagnostic testing. And that can be either prenatal or postnatal. The screening is not diagnostic. It's also important to keep in mind, and we've seen this in our clinic, that as a screening test, there's a possible outcome with diagnostic testing of having a discordant result, in which the screening identified an increased risk for a condition, and the diagnostic test identified an abnormal but different type of diagnosis. So when we have a non-invasive screen result for trisomy X, the next question is, what are our choices? What is the next step? And it's important that it be followed up with a diagnostic test that can be performed either during pregnancy as a prenatal test or after delivery as a postnatal test. So prenatally, there are two options of doing diagnostic genetic testing. One is by a chorionic villus sampling, where there's a sample of the placenta that's analyzed. And the sample is usually, it's a procedure that's performed around 10 to 13 weeks of gestation. The second procedure prenatally is an amniocentesis, where they're withdrawing some fluid from around the developing baby. And this is a procedure that can be done at 15 weeks or later in pregnancy. What we found from our research is about half the time families will defer to doing postnatal diagnostic testing, in which a blood sample or a cheek swab or a sample of saliva can be collected to do the genetic testing, and the analysis is done either the prenatal sample or the postnatal sample by conducting either a karyotype or a chromosomal microarray. So a karyotype is a study by staining the chromosomes to count them and have a total number of chromosomes, as well as and looking at the structure of each individual chromosome, making sure that there's no structural differences that are abnormal in the chromosome. So looking at a single cell, when they do a karyotype analysis, they take the chromosomes and they put them into an ordered picture called a karyotype, in which the chromosomes are in pairs. So we typically have 46 chromosomes or 23 pairs. And the pairs are ordered from largest to smallest, 1 through 22. And in each pair, we have a chromosome that came from mom and a chromosome that came from dad. Again, one from mom, one from dad, and so forth and so on. And then our 23rd pairs are labeled as sex chromosomes. And these are either given X or a Y chromosome. The Y chromosome has the genes that drive male development. And when we have a girl, if there's no Y chromosome present, then there's typically 2 X chromosomes in a typical female. But the diagnosis of trisomy X occurs when instead of just having the 2 X chromosomes, we see an additional X chromosome. And so instead of having 46, the karyotype is labeled with 47, representing that there's an additional chromosome and that it's an X chromosome in origin. A chromosomal microarray is also diagnostic of trisomy X. So in a microarray, they're using microscopic probes along the length of each individual chromosome to demonstrate the number of chromosomes present. So when we compare the first pair to the second pair to the third pair, this straight red line is representing that there are 2 copies of each chromosome. And a microarray really analyzes if there's a duplication or a deletion present. And when we're diagnosing trisomy X, what we see is there's a duplication or more genetic material along the entire X chromosome present. And it's this extra material of the X chromosome that's diagnostic of trisomy X. So the natural question is, well, how did this happen? What caused trisomy X to happen? And this occurs due to a process called non-disjunction. So when cells are dividing, either in the formation of the egg or the sperm, or if they're dividing after conception, in which a single cell at conception goes on to divide and grow into either the placenta or the developing fetus, that cell division process is subject of having chromosomes fail to separate properly, in which you end up with either an extra or a missing chromosome resulting from non-disjunction, where those chromosomes fail to separate properly. And in trisomy X, most commonly that can occur either due to the extra X chromosome in the egg or the sperm. And when we have a condition called mosaicism, where there's more than one result, not just trisomy X, but we can see other types of results in the genetic testing. This is often a result of having a post-conception non-disjunction, where at conception, the egg and the sperm had what they needed, and that the misdivision actually happened after the baby was conceived. This is not caused by something that a parent would have done either before or during pregnancy, and this is not something that is your fault. There's a lot of variability that we can see within the condition of trisomy X. And while I'm not going to go into all of the various features, because those are reserved for other talks that are being given and recorded as well, there's a lot of variation that we can see in the physical features associated with trisomy X, as well as developmental features and medical features, psychological features such as learning and behaviors. And when we look at trisomy X, in comparison to the other trisomies that we can see, XXY and XYY, we actually see a lot of overlap in some of the features. So with trisomy X, we know that there's neurodevelopmental risks that are associated with risks for developmental delays, learning disabilities, ADHD, executive dysfunction, anxiety, social communication or autism, and motor skills disorders. And we see these risks in all the trisomies, including trisomy X. From a neurodevelopmental profile, we often see more strengths in nonverbal skills, such as spatial skills, and more areas of weakness in the language-based learning or the verbal skills that we see. And then when we look at the medical and physical features, we know that girls with trisomy X tend to be taller compared to their mean parental height, or what we would anticipate based on their parents' heights. We can also see a risk for having premature ovarian failure. So this is also associated with trisomy X. We don't have a very concrete understanding, but we know that girls with trisomy X have an increased risk of having early menopause, and we want to monitor their menstrual cycles beginning in adolescence so that we're aware if this is occurring and we can offer interventions if they're indicated. And then we know that congenital malformations or birth defects are very rare, and they don't occur more commonly in this patient population compared to the general population. As far as perspectives on this condition, it's not only highly variable, but we know that family background genes and the environment in which an individual are raised can have a significant impact on an individual's outcome. We also know that when a genetic condition is identified early, that families often are given the opportunity to have a very proactive approach. And that can be from getting interventions started when they're young to having evaluations around learning that may otherwise go undiagnosed. It's really important to keep in mind that children are individuals, and individuals have strengths and they have weaknesses, and that not everything about an individual is attributed to having an extra chromosome. And we see a lot of beautiful strengths in our girls with trisomy X that we follow in clinic. From a genetic counseling perspective, I just want to make families aware that genetic counselors are also able to help with a lot of other aspects in helping you care for your daughters. So the genetic counselors often play a role in planning next steps after a diagnosis is made, understanding what symptoms may be attributed to the diagnosis of trisomy X, as well as what symptoms might be unrelated and warrant further evaluation. Genetic counselors can help with the disclosure of the diagnosis to the individual, as well as to other family members or the school if it's indicated. Counselors can help families advocate with the school in getting educational services if they're indicated and desired, as well as navigating healthcare and helping families understand how to navigate healthcare and get their child the care that they need associated, especially with the condition. Counselors can also help with the process of transition into adulthood, connecting with community resources, whether they're for children or for adults, and then addressing recurrence risks with future pregnancies or in adults with trisomy X. There's a lot of wonderful resources available through the AXYS website of genetic.org. So not only recorded talks like today's talk, as well as recorded talks from other conferences and other webinars that have been made available through the website. There's a wonderful professional directory in which families recommend various professionals across the country to other families. There's a wonderful book also published by Virginia Cover that's available that talks about trisomy X. And then our clinic and our website for our clinic, the extraordinarykidsclinic.org, has published multiple journal articles that are downloadable for families to read about the conditions and what's been published in the medical literature, as well as Amazon.com has resources that we've developed, including a series of children's books that includes Elizabeth and her Extra X. And we're getting ready to launch another series of books that'll be specific to prenatally identified babies or newborns with this condition. And with that, I'd be happy to take any questions. Thank you for your time.